This is a special episode of the Stem Cell Podcast. We're at ISSCR 2024 on day 2. Good evening, everybody. We are Doctors Daylon James and Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. Today, we're back with another special episode to discuss highlights from the ISSCR 2024 annual meeting taking place in Hamburg, Germany. If you're at the meeting, again, make sure to drop by the Stem Cell Podcast booth. It's in the exhibit hall where you can meet the team. I've been checking in there with Arun, sometimes without him, periodically, just to see if we can meet some people. It's always a rich exchange, so much fun. Today, we're going to be discussing some of the most interesting research we saw presented on day 2 of the meeting. So if you were in another session or weren't able to attend the meeting, we've got you covered. I mean, not completely covered because it's just the two of us here, but we were racing around like little rabbits, looking for sessions and looking for insight, knowledge, inspiration. We're going to kick things off in just a minute, but before we get to that,
the ISSCR's Standards for Human Stem Cell Use in Research document has been an important part of the research presented at this year's annual meeting. Learn more about the standards and get a free wall chart by visiting www.stemcell.com/ISSCR-standards. We're going to kick things off with day 2, bright and early this morning, advancing in vitro models. But honestly, I wanted to chat a little bit about last evening as well. We got a little time to experience Hamburg in different ways. I went over to the fan area of, what is it called? The fan session for the European Championships, European football or soccer championships, whatever you want to call it. Semifinals were fun. It was England versus the Netherlands. And of course, there are scientists from both countries represented here at ISSCR. And it was just a fun vibe to take part in that and watch the teams play. It was a great match. And after that, I had to grab a doner, right? That's what you do here in Germany. Don't you get a doner kebab or 2 in my case? That was a fun, delicious experience. And moving on to this morning's sessions, concurrent tracks. I went to a different one from you, but a little bit of overlap. Started things off with a friend of mine, Dave Elliott from Murdoch Children's down under in Australia. He's focused on enhancing maturation of stem cell derived cardiomyocytes and basically developing a number of benchmarks for maturing iPS cardiomyocytes by comparing their metabolically or functionally enhanced and matured tissues to primary cardiac tissues that were from a broad range of samples in a developmental timeline from a big biobank that they have down there in Australia. He was recapping their recent work, not just from his lab, but also labs of James Hudson and others, combining metabolic and functional approaches to maturing cardiomyocytes, as well as adding additional cell types to the model to make them even better. They're also, of course, applying these systems to toxicity screening, something that my lab does, looking at drug cardiotoxicity from cardiotoxic cancer drugs, for example. Moving on to the second talk, which was Helmuth Gehart. This is focusing on malignant pleural mesothelioma organoids. And I remember growing up, there was this ad I used to watch on TV about, if you're suffering from mesothelioma, please call this number and we can get you like $1 million or something like that, right? And the reason why this was so prevalent is because mesothelioma was largely caused by asbestos, which was a huge problem back in the day in terms of asbestos being a lot of building material for apartments and all that kind of stuff. Really sad situation. So a lot of people developed this lung cancer of the epithelium known as mesothelioma. And the Gehart group and their team was making organoids from the mesothelioma epithelium cells. They created a defined organoid medium for the expansion of this mesothelioma tumor biopsies, ultimately creating these organoid systems. And I think for one, I was surprised that this didn't already exist because it's such a prevalent disease and such a prevalent cancer caused, unfortunately, by this environmental situations, environmental perturbation, right? So the cool thing about these organoids is that they also reflect the overall heterogeneity of the disease. No 2 forms of mesothelioma and, cancer in general, no 2 forms of this disease are the same. So the organoids reflect that heterogeneity. Moving on to Deepti Paul from the University of Bristol. She was focusing on the leukemia microenvironment and modeling it. She was recapping a Cell Reports medicine paper that came out recently from her group that actually used an iPSC derived bone marrow system and actually a cool engineered approach that had a prototype biogel that could support the engraftment of these ALL PDX cells. And was talking about how the most critical role in this whole bioengineered construct for leukemia was the figuring out the right matrix components or ECM components in setting up this model. And they had this really nice 3D bioprinted microtissue to develop this scalable system for leukemia drug screening. Next up was Esther Frauser from Leiden, focusing on iPSC-derived inner ear organoids and also an RNA therapy for genetic hearing loss. This is a 75-day differentiation protocol, partly inspired by Carl Kohler's work. I mean, I saw she cited that work a couple times. Carl has, of course, been on the show in the past. Really nice genetic modeling of different patient-specific mutations. And then a second part that was focusing on resolving the inner ear disease phenotype with an antisense oligonucleotide therapy. And one of the questions that came up was, how feasible is this in a clinical context? And I think they still have to flesh that out, but this is a really cool model system. And the last talk in that session was actually Mary Hutch, who's focused a lot on liver and liver organoids and so on as well.
Yeah, I caught the tail end of that, although I missed the beginning of your talk, but I think what I caught, it couldn't have been much more because it was jam-packed in terms of content, as well as a very well-attended session. And I thought it was a great rendering of maybe representative of where the field is moving in terms of, are these cells within these organoids really approaching a physiological correlate? And in this case, it was really about the complexity, which we all knew that these organoids pale in comparison to the complexity of the actual liver. But I think what's been lacking is really mapping out a pathway to getting there. And here it was all about these bile caniculi network, if I'm saying that right. And a really nice rendering of the connectivity that they're able to demonstrate between three distinct cell types, all primary, of course, but still, I think a roadmap for how we may ultimately approach this in pluripotent stem cell systems driving toward liver. And also showing function, this functional demonstration of bile transport, and then finally illustration of the clinical significance with the primary sclerosing cholangitis, talking about how these tools can lend insight to a actual unmet clinical need. So yeah, I really enjoyed that story, and as I said, I think there's going to be a nice roadmap, not just for liver, but how we try and render these organoids into physiologically relevant constructs, right? Models that we can use. But going back, I mean, I got to say, first of all, I had a great night too. Loving Hamburg. We talked to a lot of people who said enjoy the weather because it's remarkably good. I don't know what that says about the normal weather here, but I had a great bike ride. I caught the game. Didn't have any doner kebab, but I got the attention of my 14 going on 15-year-old son, which is probably more precious and rare than a delicious doner kebab, I would say. So great night. And that led to the first concurrent, which blew my mind. I have a personal interest in these naked mole rats, not romantic. They're hideous, but they're such an amazing model organism for so many reasons, like all of the reasons that you would care about that are health relevant. These eusocial colonies that they exist in, which just is such an interesting, like almost philosophically, I guess you would say, they kind of have these social colonies that are akin to like bees and ants, where they have this hierarchy and there's queens and only one can be a queen. And interestingly, no one else is even reproductively competent. But the real key why there's a lot of clinical interest is because they live for like ever. They live 40 years, which is remarkable for rodents. And more than that, there's no decline at the organ level, but also at the cell level in terms of like no decline in like function, fertility, their bone density, their cardiac function. Arun, you should get into these mole rats. They're like a holy grail of heart function. But also at the cell level. And that's what this story was about from Kyoko Miura from Kumamoto University. Gave a really great, entertaining, also funny talk. Like her answers at the end to some of the questions were hysterical and insightful. But the real takeaway for me, in terms of how we could hack whatever system du jour, is that it's two mechanisms by which the cells in this organism are able to evade or have the longevity. And one, I thought interestingly, um, was reduction in necroptosis. And they showed this because even though you can expose the tissue to carcinogens, the tissue is damaged, but the inflammatory response is dampened. And that showed, and this is, and we're going to come back to this, how critical inflammation is to disease. But also showed in the mole rat, there's like a frame shift mutation in these species in RIPK3 and MLKL, whereby necroptosis is reduced. And then this was the kicker. They knocked out RIPK3, one of these genes in mice, and showed that they could delay that carcinogenic response. So they behaved closer. They didn't live forever like the mole rats, but it did at least prove in principle the mechanism. And the other thing, which is kind of the opposite that I thought was so cool, is that it's kind of has this natural senolysis. It has the senolytic properties by way of metabolism and hydrogen peroxide overexpression, where in senescent cells that would normally hang around and decrease function, in the mole rats uniquely, those are pushed to apoptosis and cell death. So it's a way of clearing, and that really, those two mechanisms, I think, alone go a long way in explaining a lot of that longevity that is evidenced in these remarkable animals. And also that this is the biggest colony that they set up in Japan. I mean, I want to get after it with collaboration with Kyoko, like now. But then it was on to a bunch of younger researchers, which I always love to see. I got a session from Danielle Posterak, who's a Merit Award winner from Netherlands Cancer Institute. And this was like this Rambo epitope barcoding they called polytope, which is like confetti to the next level. 9 different epitope tags that could lead to 512 different color codes doing lineage tracing. There's a lot of use cases that were shown in the gut, embryonic brain, T cell development in the thymus, cancer, all really big targets. Fate mapping with clonal dynamics, highly quantitative, really a tool that I'm sure you're going to see in a big publication. We'll probably cover it on the show. Then it was on to Duncan Chad Chady. Sorry if I'm mispronouncing their partner. This is a grad student from Caltech who gave a great, amazing, well delivered talk about another lineage tracing strategy. In this case, recording cellular histories with this CRISPR AG base editing. Arun, you've talked about base editing and all these different CRISPR analogs and derivatives a lot on the show. In this case, it's just, you make this super mouse with a big template of double A's, and then you get 3 outcomes. I mean, you're going to see this paper out too. This is a nice inducible system where you can get a lot of different outcomes and do lineage tracing on the next level. I also caught that Esther Fauser paper that you covered, so I'm not going to go over that other than to say that the charm there for me, this is a travel award winner from Leiden, who was 2 years into her PhD and on a big stage giving a talk that you described really well, so I'm not going to go over it. But other than to say that her poise was remarkable, and also her enthusiasm was palpable in describing like the real, I mean, not to throw any shade on it, this was like the most advanced first 2 years of a PhD that I've ever witnessed, obviously, to get to the level of giving a talk at the ISSCR, but it was still like a lot of fundamentals. And you could really see how excited she was by every result. Like I could see the reflection. She took a picture of her gel and I could see the reflection of her face in the computer screen, and she was beaming and I wanted to cry. And yeah, like I said, we ended with Mary Hutch, which was a great session in terms of like what we need to get to in terms of these organoids. And then it was quickly into we had a short break, and then it was into plenary 3, which I think you could cover when you were there, right?
That's right. Yeah. So plenary 3 was led off with a little bit of housekeeping from 2 guys that I know pretty well. One, Clive Svensson, obviously my boss at Cedars Sinai. I've got to say nice things about him, right? He's the ISSCR treasurer and gave a good overall impression of the finances of the ISSCR. They are, quote, great in his words. Also, the overall growth of the society, and also making sure that the eye in ISSCR is emphasized, and that it's truly an international society, and that the membership of the society really reflects that. Keith Alm was next up with the housekeeping. He's, of course, a CEO of the ISSCR, and he gave various notes of thanks, especially to his support staff. And he's been on the ISSCR staff for 5 years as well, helping with the growth of the society. Also gave a big thanks and shout out to Hans Scholler for making Hamburg happen. Like what we talked about yesterday, this meeting has been close to 8 years in the making and was delayed a bit thanks to COVID. But now it's here, and it's been such a fantastic meeting so far in many different aspects. Keith also gave a big thanks to Amanda Clark for not just her presidency, but for 20 years of supporting the society. Martin Para was up next. He's, of course, the editor in chief of Stem Cell Reports, but soon to be stepping down from that role to let Janet Rossent actually take over as the new editor in chief. That's pretty exciting. He also gave a shout out to a new initiative where they're able to reduce publication fees by 50% for first time submissions for ISSCR members to Stem Cell Reports. So be sure to check that out. I published there a couple of times. I love that journal, honestly. And then moving into the meat of the science for the plenary, first off, Jennifer Nichols investigating the mystery of human trophectoderm expansion. This is a great look back over the years and her lab's work on advances in early trophectoderm modeling, answering this question of whether human epiblast cells can regenerate trophectoderm. She does show some really cool fragment cultures of pluripotent stem cell derived early embryo models, nice labeling using these fluorosphere systems to examine the early trophectoderm dynamics. Really just nice overview of that particular field. And she's, of course, one of the world experts in that area. Some statistics as well I thought were quite striking that showed how hard it is to actually culture early human embryos ex vivo and actually successfully derive naive pluripotent stem cell lines from these embryos. And finally, some blastoid models of human trophectoderm abnormalities as well. Second talk was Marina Elena Torres Padilla from Munich. Epigenetic mechanisms of cellular plasticity and reprogramming to totipotency. Of course, this is the most powerful state of potency in stem cells, toti meaning giving rise to anything. And pretty much the only cells that can do that are the very, very, very early stage embryos. We're talking about two cell embryos, two cell stage, as well as two cell like cells or two CLCs, which mimic the cells comprising the two cell embryo. Can we drive or induce totipotency? Sort of analogous to what you see with the Yamanaka factors. And there's been a lot of really exciting work in this area recently, figuring out the right cocktails of factors that can maintain this two cell like state for a long period of time. And also figuring out and conducting a high throughput screen to identify low doses of retinoic acid, surprise, surprise, as a robust inducer of the two cell like cells. Really neat study there. Moving on to Mansi Srivastava, which was one of my favorite talks, maybe my favorite talk in this whole ISSCR so far. It's only day two, right? Exploring the pluripotency of stem cells during development. And she's at Harvard, really establishing an amazing model system with these ACL worms, aka the three-banded panther worms. It's a unique model system which has multiple adult populations of pluripotent stem cells. And we've covered other worms, like the flatworms, for example, that have the same incredible regenerative capacity of basically you can cut off a portion of the worm and it's going to grow right back thanks to these amazingly powerful adult pluripotent stem cell populations. I'm a fan of the cool model systems. You know that. There's lots of cool tools that they've actually developed in their lab to interrogate the genetics and the function in these worms with these Pee-wee positive stem cells that are involved in regeneration, not just in this species, but in multiple species. I mean, the one of the really important questions that they're trying to answer is that how are these super powerful adult stem cell populations made in the first place? And they did some pretty cool lineage tracing with these transgenic reporter lines. Really beautiful system and very, very powerful in studying regeneration in the adult system. And then finally, we had the Anne McLaren Memorial Lecture, Pluripotent Stem Cell Models of Gastrulation from none other than Alfonso Martinez Arias.
Yeah, I mean, he's been really outspoken about this subject, and this, of course, was the Anne McLaren Memorial Lecture, and I thought he took a real, it was a nice touch, a great approach that he took in titling his story "Drawing Lines," which was a callback to this 1984 paper by Anne McLaren, where she, discussed and explored and really, I think, established a template of the so-called 14-day rule, but really with the spirit of just like asking questions and really trying to explore these ethical boundaries in an agnostic way, I'd like to emphasize, and in a scientific way with a lot of caution and consideration. And Alfonso, I think, as I said, in a great touch, didn't present data. He just did a really nice review, an homage, you might say, to Anne McLaren, her foundational contribution to IVF and the study of pre-implantation development, but also as a trailblazer, considering all of the tough ethical implications that she faced at the outset. Remember, her contribution predated and perhaps established a pathway towards IVF, which has been a tremendous clinical success. You might say the greatest clinical success in advance of the last 40 years in terms of solving an unmet need. And nowadays, it's really highly relevant considering the proliferation of embryo models and forcing us to revisit some of these tough questions that are always going to come back when you're working with something so important and so visible with such a great upside, but also a lot of negative attention and target assigned to it. So I thought a really insightful review, handled delicately, but also courageously, so saying what needs to be said. So I really enjoyed that. I learned a lot, and it made me think, and it made me hungry. So I took Arun out for his second doner kebab in less than 12 hours, and he was not complaining. We found a good one. A little shout out to Hamza Kebab, where we got a beautiful doner in a box just to keep it clean because we're trying to present ourselves well for all you people out there. While we're still at the conference, late night though, I'm going deep on that Philly cheesesteak style doner that you had last night. I remember that thing was devilish.
Delicious.
Ah, well, let's reel it back from my future delight and take it to another one of my scientific delight in plenary 4, which was tissue architecture. And it jumped off with Krista Lo Celso, sorry for the mispronunciation there. And this was nice, close to my heart in that I love stories that talk about, in health and disease. And this was one of those talking about leukemic cell regeneration, but also, normal, a healthy hematopoietic stem cell survival and recovery, both sides of the coin there. And really the common denominator being the role of inflammatory stress and how inflammatory stress can actually drive leukemia cell regeneration, but also on the flip side, how hematopoietic stem cells that can survive inflammatory stress can go on to be healthy and contribute significantly to reconstitution. So a really nice, important contribution there. Then it was on to Heinrich Jasper, who's actually in the States. He's a Genentech head of, I think, cell science, stem cell science. I don't know. But I works in Drosophila. Wouldn't have guessed. Not familiar with his work, but he's been amazingly productive. It's surprising that I haven't heard about his contribution already, because he said, like, in a little bit of a, I'd say, a modest flex. He was like, "Yeah, I'm just going to talk about my academic." He's like, "I'm just going to talk about my academic stuff. No big deal." And then he proceeded to string out like 100 papers. So it was all, I think, mostly linked by this the Drosophila as a model and stem cell response to aging, but also drawing a line to like the human lung, injury recovery and homeostasis, the maladaptive role of PTK7 mediated repair there. So that was a revelation that they're still doing basic science at Genentech, number one. And two, who knew they were so good at it? a great series, I'd have to say, of stories, all published. I would advise you guys to go check out Dr. Jasper's body of work. And then it was on to Fiona Deutsch, a legend, of course, who's at Basel, studies adult neural stem cells. And I mean, this is where I know I'm an old man, but also it shows how fast the field is moving. When I started in this game, we still thought that there was no new neurons made in the brain. We were like, yo, it's static, that's it. And since then, of course, we've learned a lot, and a lot of it from Fiona Deutsch. Here, she described about how the subventricular zone, the site of neurogenesis, is actually innervated by multiple neural inputs that regulate neural stem cell production like on demand. And then went deeper and talking about in the context of pregnancy to kind of endow that concept with some relevance. And in this case, showing that's a key to recognition of like your own pups. So I just thought that was really cool, lent credence and gave new meaning to that whole term of pregnancy brain, which anyone with a partner or who themselves has had a child may be familiar with. And then it was on to the incomparable Elaine Fuchs, who has not only been like, she gave a talk. I'm sure she gave like a few sessions moderating, running, talking, good. But she also, what I love about Dr. Fuchs is she gets up and asks really good questions, which shows like she just knows everything. She has an encyclopedic knowledge across the board. And it's probably how she's so productive, right? She feeds all that interest and passion in other fields in the stem cell universe, and then kind of ties them back into her own line of research. So she gave, Elaine's gonna Elaine, and she went up there and talked about a series of science that she's produced, and probably in the last couple of months. She's got like 7 Nature's papers on deck. Not really, but she's tremendously productive. And I joke, but she continues to reinvent her science, in this case focusing on like inflammation, and I think that was a common theme here, a little callback to maybe where the field is going, recognizing and appreciating, not that it's new, but emphasis on inflammation and post-inflammatory recovery and behavior of stem cells, in particular in multiple systems. It was a great talk. I had to cut out a little early so I could come into the delegate session and interview. We ended up talking to 12 people in the last couple hours before doing this roundup. So I mean, I have to say, I've had my favorite talks. I'll wait to the end to share what my favorite was, although I did love that panther worm. That thing was dope. But talking to the delegates, I guess we're calling, was my favorite part of this meeting so far. So much fun, Arun, to meet the faces, and to like just hear about how interesting this next generation, for the most part, it's younger scientists, how this next generation is charting the course. What do you say?
Yeah, it was really fun to chat with a range of folks from junior trainees, PhD students to some group leaders as well. It was a recurring theme. And we're not going to spoil the episode for you. It's going to come out in a few weeks, but one of the questions that we asked was, in the future, where would you like the ISSCR annual meeting to be held? And the dominant answer actually was Japan. And I don't disagree. Japan is one of my favorite places in the entire world, has such a deep stem cell history and recent stem cell history too, with, Dr. Yamanaka and the iPS revolution. But also Australia was very well represented on that list, and India showed up as well. So honestly, we'll find out pretty soon, I'm sure, where 2026 ISSCR is going to be held, and can't wait for that meeting.
Yeah, but if that steering committee is listening, everybody, like literally everybody, is thinking Japan. So make a note. That brings us to the end of day 2 at ISSCR 2024. Don't forget to follow us on X at STEMCELL Podcast to find out what we're up to at the meeting. We're constantly updating the social. We're all over the place, running around, eating some doner kebab in like pretty much every institution that makes it. Visit us also at the STEMCELL Podcast booth on the exhibitor floor, where you can win some prizes. We got prizes. We also got freebies. You don't have to win. It's like very generous. They're very generous. Check back here tomorrow also for another episode recapping day 3 of the meeting. Until then, thank you guys so much for listening to this episode. We'll be back at you sooner than later.